Great, thank you so much. I'm really happy to be here today, and thank you all for coming, especially those of you who are coming in the middle of the night for this. I appreciate that very much. So yes, today I'm going to jump in and that when we look at the animals that dominate, host chemosynthetic bacterial symbionts that provide the bulk of their nutrition. Let's move this forward. And so these symbioses, they're obligate for the animal, and so the animal needs the symbionts for their nutrition. And so in many cases, these symbionts are living inside the cells of the animal, where they're using the electron donors from vent fluid, like the hydrogen, the sulfide, and the methane, pairing that with the electron acceptors that are found in just the surrounding seawater, like oxygen and nitrate. And that provides chemosynthetic energy to fix inorganic carbon into organic carbon, which is used for the symbiont for its own biosynthesis and growth, but also provides primary nutrition for the host. So these are obligate symbioses that the host needs in order to survive. So when we think about these obligate symbioses, you can imagine that it's necessary for the host to have some system of reliable intergenerational transmission to their progeny. And so there is one way of doing that, and that is through this idea of vertical symbiont transmission, where the symbiont is directly passed to the progeny, typically through the maternal germline. So you have reproduction, the symbiont is in the maternal germline, and then progeny end up with the symbiont directly from their host, for the parent. And I'm just bringing this up now because it will become relevant in a couple of cases that we bring up with the data later. So when we have vertical transmission systems, we see commonly these patterns of co-diversification, where the evolutionary histories of the host and their symbiont match because they're paired together, they're tied together physically and over evolutionary. These phyla are sort of a visualization of their evolutionary history where they're congruent, where they look pretty much the same. And so when you look at these together, you see these pairs are sort of matching one-to-one based on their phylogenetic history. So we also have another method of symbiont transmission that exists, and that is something called horizontal or environmental symbiont transition. And that is where the host has an aposymbiotic stage, and the progeny must acquire the symbiont from the environment in order to survive. So you have production, their progeny do not have a symbiont, so they're aposymbiotic, and then they have to take up a free-living symbiont from the environment, which has either been seeded by the adult host during its life, or perhaps during the death of the adults in that area. And so when we see this type of transmission, it does not always result in co-diversification, where we instead will see a mismatch of the evolutionary histories of the host and the symbiont, where they're independent from one another. And so we don't see that these are this sort of they don't correspond perfectly and mirror each other. So I'm just bringing these up because they'll be useful later when we look at some data. So in the ocean, including at hydrothermal vents, horizontal transmission of symbionts is common. And this is facilitated by the aqueous medium, which is water, where symbionts can sort of persist in the environment and be transmitted through the environment that way. But horizontal transmission has risks. So this is really interesting. It's very common, but it has risks. And so this is the risk that the progeny do not find their partner, their symbiont at all, and so then therefore cannot survive in the case of an obligate symbiosis, like the ones at vents. Or that you might accidentally take up a cheater or a harmful microbe like a pathogen instead. So there are risks to this transmission system, and yet it seems to be the most common in the marine environment. And so there's been this long-standing paradigm that the risks of horizontal transmission have been balanced by the fitness benefit of associating with a locally adapted habitat-specific symbiont strain or species. So, where you have a host that might go to a new environment and then take up a free-living symbiont in the new environment that is sort of locally optimal or locally adapted to that environment. So that's sort of the paradigm that has existed for why we commonly see this in the marine environment. And so this is especially important in symbioses where aposymbiotic larvae disperse over long distances to new habitats or to habitats that are variable over time. And so vents are a perfect example of this, where you have larvae that can disperse long distances to new vent fields, or if you do see recruitment back to the original site, that site is highly variable, even within a single site and then over time as well. So the ability to sort of pick up a symbiont that's specific to the habitat that your progeny will end up in seems like it could have a lot of benefit. And yet, at least for the vet environment, we don't have a lot of direct data looking at this for most symbioses. And so my lab has been working on sort of addressing this paradigm in a group of hydrothermal vent snails. And so in general, what I'll be talking about today is some questions that we've been asking about how these hosts transmit their symbionts to their progeny. And then also looking at whether these hosts associate with locally differentiated symbiont populations. And if these symbiont populations are different, depending on geography, do we see evidence for local adaptation or functional differences between symbionts at different sites? And so we've been working on two sister genera of hydrothermal vent snail that are dominant in the Indian and Western Pacific Ocean hydrothermal vents. There are 6 species in the genus Alviniconcha, and those are shown here. And then they're colored by their different species here on the map, so you can see their distribution, as well as their a single species from their sister genus Ifremeria nautilus as well. And so I just also want to note that these species are all considered endangered or vulnerable, and that's due to the threat of mining in the regions that they occupy. Okay, so in the case of Alviniconcha as well as Ifremeria, we see these animals clustering around hydrothermal vents. So this is an image here of diffuse flow hydrothermal vents with Alviniconcha clustered around. And they're doing this in order to access the chemical reductants in the vent fluid, the sulfide, the hydrogen that's coming out of the vents. And that's because they host chemosynthetic bacterial symbionts in their gill tissue. So this is an image of an Alviniconcha gill. And so the blue here are the host nuclei, and then the yellow here are all bacterial cells. So they're packed inside the gill tissue of these snails. Okay. Oh, sorry, there's an animation here that I didn't want to come up. Okay, so we've known for quite a while that Alviniconcha and Ifremeria associate with 7 species or genus level, what we call phylotypes of symbionts that come from 2 bacterial phyla. And this is, was work that was pioneered by Suzuki in 2005 and 2006. And then since then has been corroborated by myself and others, seeing that we have these very specific relationships between different hosts and particular symbiont phylotypes. And so when we look at this broadly, we have this conceptualized host phylogenetic tree, and then the conceptualized symbiont phylogenetic tree here. What we see is that there's not this evidence of co-diversification. And so it's been long hypothesized that Alviniconcha and Ifremeria transmit their symbionts horizontally through the environment. And this is the primary evidence for that. Okay, so we recently embarked on a study that's a sort of global collaborative study looking at the symbionts of Alviniconcha and Ifremeria across their distributional range. And so this is some data that's in a paper that's currently in review, being led by a postdoc in my laboratory, Dr. Corinna Breising. But it's a collaboration with Didier Jolivet, Thomas Broquet, Peiwan Qian, and Jin Zun as well. So it's a global collaboration where we've all contributed samples for this analysis to look at the symbionts across their distributional range. And so we end up with 761 snails total in this data set. So it's quite a large data set. And so we performed amplicon sequencing on all of these individuals and found that we did consistently see the same relationships that we had been previously seen with these species, where each species sort of has specific symbiont phylotypes it associates with. But interestingly, when we actually look at this at the strain phylotype, represents 2 to 23 strains or species level lineages. So there's some diversity within each of these phylotypes when we look at this broad scale. And so again, what we confirmed with this is that the symbiont associations are specific to host species, with the exception of 2 Alviniconcha species, Alviniconcha Kojimai and strummeri, which overlap. So again, here we're coloring the different snail species here, but with different colors. Plot showing the in. You can see that they're clustering by species fairly nicely, except for in the case of Alviniconcha Kojimai and strummeri, which overlap in their symbionts. So again, we're confirming with this large-scale data set that these symbionts are specific to their host species, at least at the level of phylotype. We also find with this data set that an individual snail is typically dominated by 1 symbiont phylotype, and then there's sort of 1 to 2 what we call oligotypes or strains present within an individual. So here we're showing again the relative abundance of these different symbiont phylotypes at the strain or species level, and then each of these bars is an individual snail. And so you can see that in most cases, they're dominated by 1 color, maybe 2 colors, but these are primarily from the same symbiont phylotype. So they're usually dominated by 1 phylotype, but have some sort of strain level diversity within an individual. And so what we also found is that when we look within a host species, we do see some subpopulation differentiation according to geography. So again, what we're looking at here is the symbiont composition. And in this case, this is just showing an example of Alviniconcha Kojimai, one of the species. And then here now we're coloring by different sites where the individuals were sampled from. And what we see is that there is clustering by color, which is by geographic site. So we are seeing that these, even though there is specificity at the species level, there is sort of this strain level variation where we see geographic subpopulations that are differentiating from one another at this large scale. So when we see this, the question is this difference or this differentiation and population structure driven by differences in host genetics? Like, are there differences in the genetic of these hosts from these different sites? They have subpopulations, and that what may be driving the structure we're seeing. Does this have to do with neutral processes like dispersal barriers to the symbiont? Or does this also have to do with adaptive processes like adaptation to specific habitats in these different geographic regions? So we then embarked on a sort of smaller scale study to address these questions in more detail. And this again was led by Corinna Breusing, the postdoc in my lab. And in this case, we were focused on Alviniconcha and Ifremeria from the Lau Basin, in sort of near Tonga and Samoa and Fiji. So this here shows 6 vent fields separated by up to about 700 kilometers in the Lau Basin that we were able to sample these individuals from. And so this is a great place to look at sort of the relative contribution of the host genetics and environment because of for a couple different reasons. First is that for the host, this is an area that's predicted to have well-connected host populations. And so based on what's known about the larval biology of Alviniconcha and Ifremeria, which is not much, and so that's why I have an image of another gastropod larvae here, not of Alviniconcha or Ifremeria, because they're not particularly well studied. We believe that these animals are likely to have long dispersing planktotrophic larvae that could disperse long distances. And then through a nice dispersal model published by Mitochondrial in 2016, there was no barriers to dispersal predicted to be exist within this basin. So these should be sort of well-mixed host populations. And this is also a great place to study the effect of environment on symbiont composition, because this is an area where we see habitat conditions that vary within the region. So for example, if we look at the Eastern Lau Spreading Center, which runs sort of north to south here with a number of sites distributed along that, we see a large change in geology from basaltic to andesitic hosted vents from north to south. And this corresponds to a change in the fluid chemistry, where you have higher concentrations of hydrogen sulfide and hydrogen in the north and lower concentrations of these two things in the south. So this is a great place to look for the effect of environment on symbiont population. Okay, so first we wanted to examine, again, idea of horizontal transmission in these species. And so to do this, what we did is that we assembled host mitochondrial genomes and symbiont genomes from the same snails, and so we had host symbiont pairs for these 4 different species, 3 Alviniconcha and Ifremeria, and then their corresponding symbionts that they associate with, and we had a number of symbiont pairs from many different sites in this region. And so then what we could do is we could compare their phylogenies to look for evidence of co-diversification. And in all 4 cases, we do not see that the phylogenies were congruent, suggesting again that this is these snails use a horizontal transmission system in their symbionts. Okay, so when we wanted to go further then is to look at the symbiont population structure and how that varies within the single host population, and looking for evidence of what was driving this variation. Okay, so to do this, we did a population genomic study of both the host and the symbiont. And so we first started out by taking those symbiont genomes we had assembled for each of the different symbiont types and constructing what's called a pangenome, which is basically where we combine a number of symbiont genomes into sort of one genome that represents the population. So we had a pangenome for each of these 3 symbiont types in the region. And then we also constructed host transcriptome references from some previously published Illumina RNA-seq data for these 3 Alviniconcha species in Ifremeria, where we co-assembled a number of individual transcriptomes, ended up with reference transcriptomes that were from 30 to sort of almost 60% complete. And so we could use all of these to do our population genomics work. So then what we could do is take individuals of these 4 host species from these vent sites, and we can sequence using Illumina sequencing the gill tissues of these animals, which is a mixture of both host and symbiont reads. And then what we can do is we can take the reads from all of these individuals and compare them or map them, the reads, to these references. And so we have the symbiont pangenome as a reference for the symbiont populations, and then the host transcriptome references as well. And this allows us to assess population level genetic structure, basically by finding sequence variation between individuals. So this could be single nucleotide polymorphisms or SNPs, or insertion deletions, or also the presence and absence of different genes in these populations. So to start out with the host data, it looks like it's a little cut off there, so I apologize about that. So what we found is that there was, within each host species, we did not see any genetic structure by site. So this again suggests that these are sort of single populations across all of these sites in the Lau Basin. And again, this is consistent with what was predicted based on larval biology and dispersal models. So this was not surprising to us here. So basically what we're looking at here are plots showing the genetic structure of each of these 4 species. Their color, each of these individual points is an individual snail, and they're colored by site. And so what you see is they're well mixed. All the colors are sort of mixed together. There's no structure or clustering, which again suggests that there's no genetic structure by site in this area. Okay, but when we looked at the symbiont data, what we found is something as a sort of a picture that was quite different, where we did see structure by site. So here we have Alviniconcha boucheti and its particular symbiont phylotype, which we call epsilon. We have Ifremeria nautili and its particular symbiont. And here we're looking at is sort of symbiont population structure, where each of these individual points is an individual snail, and it's colored by site. And so you can see that there's clustering by color, which indicates clustering by a geographic site. When we look at Alviniconcha Kojimai and Alviniconcha strummeri, these are the species of Alviniconcha that I mentioned earlier share a symbiont phylotype. And so this is one we call gamma 1. What we found is that there is a differentiation by host species. So here, each of these individuals is shaped by the species, where the circles are Kojimai and the triangles are strummeri. And so you can see that they are separated from one another, indicating that they host distinct subpopulations of this particular symbiont phylotype. But we also see, even within those species, clustering by geography as well. So there is, in this case, strain level population structure between site and also between species as well. Okay. So, so far, when we think about these original questions that I had posed at the beginning, what we find is that, how do these hosts transmit their symbionts? We don't have evidence for genetically separated host populations, but the symbionts show genetic structure by geography, and so this, along with the lack of co-diversification in the phylogenetic trees, indicates again that we're confirming this hypothesis that they have horizontal transmission system in these snails. And then, this question of do hosts associated with locally specific symbionts? Yes, it does suggest that the symbionts are being acquired from a locally distinct environmental population after dispersal. So the hosts are genetically well mixed. They appear to be able to sort of distribute throughout the Lao Basin, and they seem to be associating with this particular symbionts that are present in their phylotype at a particular site that they arrive at. Okay. So as I mentioned, the other thing we wanted to look at was the potential for adaptation and functional differences in these symbionts by strain according to site. And so what we did is looked for first at the presence absence of different genes in the genomes of these symbionts according to site. And what we did find is that there is some differences, consistent differences in the gene content of these symbionts strains according to site. And so a lot of the variation that we're seeing does relate to energy sources that vary in their concentration, for example, hydrogen and sulfide. So here what you're seeing are the genes that vary between sites in each of these different host species and their particular symbiont at the top here. So each of these columns is an individual snail. It's colored by the site that they came from. And then each of the rows is a different gene. And so if the gene is present, it's dark blue. If it's absent, it's light blue. And so you can see that there are many genes that are present in the strains from one site, but absent in the strains from another site. And a lot of this variation shows up in genes that are related to chemosynthesis, which is really interesting. And just to go into more detail, and perhaps one of the most interesting stories is that we found that when we look at this gamma-1 symbiont type, that strains from the most southern site, which is Tui Malila, are missing many genes for hydrogen oxidation compared to the strains that are found in the northernmost sites, which have higher hydrogen concentration. So we have here, all of these genes are present in the strains in the north, where there's more abundant hydrogen in the hydrothermal fluid, and then they're missing in the strains from the south where hydrogen has a lower concentration. And this was a really interesting observation because it made us sort of rethink some of the experimental results that we had published previously. So this is work from my lab, again, led by Corinna Breusing, a postdoc in my lab, where we did some high pressure experiments measuring chemosynthesis with hydrogen in these different Alviniconcha species and their symbionts. And so what we found is that there was some variation within these species here, Kojimai and strummeri, where we see, these are different experiments with animals from different sites. And we saw there was quite a bit of difference in their ability this is their rate of carbon fixation using hydrogen in our experiments. And so you can see in some cases their chemosynthesis was much greater than the other experiment. So when we looked back at these, what we found is that these experiments where we had higher rates of chemosynthesis associated with hydrogen were with animals that came from these northern sites with the higher hydrogen concentration, whereas the animals that had the lower chemosynthesis rates with hydrogen came from the southern site with lower hydrogen concentrations. And these are areas where we have strains that are actually missing many of their hydrogen oxidation genes. So what we're saying is that this variation in gene presence and absence may actually be showing up in our phenotypic assessment of chemosynthesis in these snails. Okay, so What this shows is that there does appear to be strain-level symbiotic variation in gene content relating to chemosynthesis, which is corroborated by our previous phenotypic experimental data. What I didn't show today is that we also did some work looking for signatures of selection on shared loci, which also suggests that some of the geographic variation we're seeing is adaptive. So what we're seeing is some evidence that these different geographic strains may actually be functionally different as well. I just want to note also that this is a paper, this data all came from a paper that is all currently in review, and we do have a preprint available for this work as well. Okay. All right. And so I wanted to move on to talking about the free-living symbionts in the environment, since now we've provided evidence that these Alviniconcha and Ifremeria snails are transmitting their symbionts through the environment. Now, you might be wondering, what about the free-living symbionts in the environment? What are those populations like? So I'll be presenting some work today by a graduate student in my lab, Michelle Hauer, looking at free-living symbionts of Alviniconcha. And so this is a different study that is also currently in review, and this is a collaborative study with Julie Huber at Woods Hole and Elizabeth Turnbuck-Reichert, who's now at Arizona State University. And so in this work, what we did is we compared the symbiont populations associated with Alviniconcha hesleri, and that while they're in the tissue of the snails, as opposed to the symbionts in the environment. And so this work was done in the Mariana Back Arc Basin at two hydrothermal vent sites, Hafa Adai and Ilium. And at both of these sites, we collected Alviniconcha hesleri from these sites. And then Julie and Elizabeth collected free-living microbial samples from right around those Alviniconcha snails. So this is an image of them sampling hydrothermal fluid right over a big snail bed of Alviniconcha. Okay, so from those samples, what we did first is we assembled symbiont genomes from those samples. And so Elizabeth and Julie had previously assembled symbiont genomes from their free-living metagenomes that they had done for a separate study. And so they had observed that there were symbionts present in those metagenomes. And so they had assembled symbiont genomes from the environment. And then we took animals from those very same sites and assembled the symbiont genomes that were associated with the host tissue. And so when we ended up with 4 symbiont genome assemblies, that were either from free living, that's showing in red here, or blue, which came from the host tissue. And so this is a phylogenomic tree of the symbionts. And so my main point in showing you this here is that these all cluster together, and they cluster by site and not by their sort of lifestyle, free living or host associated. And so when we look at the average nucleotide identity of the genomes, the pairwise identities are all greater than 99%, indicating that these are strains of one bacterial species. So we have these 4 symbiont strains from these different lifestyles and different vent fields. So then with these symbiont genomes, we were again able to create a symbiont pangenome. So again, we have these 2 symbiont genomes from the tissues and 2 symbiont genomes from the environment. Together makes one pangenome that we can use for population genomics. And again, it was very similar to the previous study that I presented in the Lau Basin, although here we have a much smaller data set. We have 6 snail tissue samples and 3 free-living samples from the 2 vent fields. And so again, we sequenced the gill tissues and the free-living microbial communities from the vent fluids, and then with Illumina. And then what we can do is take the reads we get from those samples, compare them to the reference, which in this case is the symbiont pangenome, and again, assess population level genetic structure between these samples. So I wanted to at least just start out by pointing out that we the symbionts, are present in the hydrothermal fluid. Compared to the gill tissue, it's a lot less of the reeds that are returned come back as symbiont reeds. So in the gill tissue, it can be up to 40% of the sequence reeds are from the symbiont, whereas in the hydrothermal fluid, we're less than 1% of the sequence reeds. But they are present, and we can use them for this analysis. And then I also wanted to point out this calculation we did to try to get a better sense of what we were seeing in the environment. There was some suggestion by others that, that you could have free-living symbionts in the environment that are actually free-living outside of host tissue, or when you're sampling the environment, perhaps what you're doing is sampling either larvae that might have some symbionts attached, or maybe more likely that you would have shed gill cells where you have they're being sloughed off into the environment. And so one way we addressed this was by calculating the symbiont, there should be 16S here, symbiont 16S rRNA gene versus host mitochondrial CO1 gene ratio in our samples, and compared that in our gill tissue versus our hydrothermal fluid. And so in our gill tissue, the ratio of symbiont to host genes was up to about 6, whereas in the hydrothermal fluid, it was up to 242. And so what we think this indicates is that, at least some of the symbionts we're capturing in the hydrothermal fluid are living outside of the gill cells, because we would expect these to be the same or very similar if what we were sampling were just shed gill cells. Okay. So when we look at the symbiont population structure, again, what we're seeing here is the symbiont composition population structure by site, and so it's colored by site here. We have 2 at Hapa Adai and 1 at Ilium, and then we have they're shaped by either free living, which are the circles, and host associated with their triangles. And so what we see is that there is a population structure that is more strongly or more strong population structure by geography than by lifestyle. So you can see the separation here. Along this axis is the separation between Hapa Adai and Ilium. And then, we do have this one free-living Hapa Adai sample that's separating from the other Hapa Adai samples as well. But still, we see the greatest sort of segregation is along this axis, which is geography. So again, what we want to look at is the potential for functional variation in these populations. And so we again examined symbiont gene content and saw that it's varying again by geography, not by lifestyle. So again, you're seeing that these plots that show the presence or absence of genes. Each of the rows is a different gene, and then each of these columns is an individual snail. Here I'm showing the site that they came from, and then the shape shows whether they were free living or host associated again. And so we see that there's separation in their gene content by geography. These are the Hapa Adai ones on this side, and these are the Ilium on this side. And then again, they seem to be sort of the main sort of pattern we're seeing is by geography and not by lifestyle. And so when we actually look at the genes that are varying in this case, we see that they vary in a number of different gene categories, but there's some notable variation in genes involved in the transport and metabolism of metals, for example, cobalt, zinc, cadmium, and iron, as well as interaction with phage. So unfortunately, there's not a lot known about the metal concentrations in the fluids at these different sites, so we can't really directly link what we're observing here with sort of known environmental variation, but maybe in the future we'll be able to do that. And then I also want to note that this is an interesting observation that the genes involving how these symbionts are interacting with phage suggests that that phage may be an important part of geographic structure in these symbiont populations. Okay, so again, just to summarize, the Alviniconcha symbionts are present in the environment, and they're more similar to host-associated symbionts from the same site than from free-living symbionts from another site. Okay, but just to summarize at the end here, sort of the main points that we found is that Alviniconcha and Ifremeria symbionts are likely transmitted horizontally through the environment. So they're acquiring their symbiont at some aposymbiotic stage from the environment. We don't know what that stage is for these snails is yet, although that's something that we're continuing to work on in collaboration with some larval ecologists. What we also found is that Alviniconcha and Ifremeria symbiont phylotypes are species specific, but these snail species can associate with a range of strains or sometimes species within that phylotype. So, you know, they do seem to be specific where they have their own particular symbiont, but they have some flexibility, often at the strain level, sometimes at the species level in that particular phylotype. And then we are also building some evidence that Alviniconcha and Ifremeria associate with locally specific and possibly locally adapted symbiont strain species according to geography. So, you know, the evidence we're building is genomic. It's not directly phenotypic data. And so, you know, future work, perhaps using experiments to sort of test their functioning in different environmental conditions will help us understand this better. But the genetic work does suggest that they have functional differences that indicate they're locally adapted. Okay. And so I guess broadly, when we think about the ecology of Alviniconcha and Ifremeria, thinking about this balance between symbiont flexibility, which has the advantage of local adaptation and, you know, perhaps enabling a broad geographic range, versus symbiont specificity, which, you know, they need some amount of specificity so they're not just taking up any old bacteria from the environment and getting a pathogen or a cheater. So this sort of balance and understanding this balance is going to be really important to Alviniconcha and Ifremeria and their ecology. So this is again from the Rhode 2016 study looking at the dispersal of these snails, or the potential for dispersal organisms in these snails ranges. And you can see they have the potential for sort of wide-ranging dispersal. They have broad ranges. And so some of this ability to have these populations be connected and to have these broad ranges may relate to sort of this balance between flexibility and symbiont specificity. And then I'll just end with making the point that, you know, while the data I presented today is specific, of course, to these particular snail species, you know, given sort of that many dominant hydrothermal vent animals are symbiotic, and many of them are thought to transmit their symbionts horizontally, the sort of balance between symbiont flexibility and specificity is going to be generally important to our understanding of vent ecology broadly. All right, so with that, these are my wonderful lab members and collaborators and the sort of the crew of the ROVs and vessels that we've used to do this work, as well as my funding sources. I have my contact information down at the bottom, so please don't hesitate to contact me to discuss this more. And then the last thing that I'll say beyond my acknowledgements is I just wanted to put a plug in for this new aquatic symbiosis genomics project that I and many others around the world are part of. And so with this project, the 6 Alviniconcha species and Ifremeria nautili, as well as their symbionts, will all be sequenced by the Sanger Institute in the UK. This is a sort of collaborative project funded by the Moore Foundation with the Sanger Institute. And so, you know, we will have these genomes forthcoming, which will be very exciting. And all this data will be publicly available for anyone to use. And I also will note that there are other vent mollusks and tube worms that are part of this project broadly as well. So exciting for vent symbioses in the near future. So with that, I'm happy to take questions, and thank you all very much.